Bismillahirrahmanirrahim. We start another paper 4. This is now the June 22 paper 4-2, and this is the first video, which is question 1, 2 and 3. Now coming to the question number 1, the jaguar whatever is the large cat that lives mainly in South America. The majority of jaguars have light brown fur with black spots as shown in figure 1.1. Some jaguars have completely black fur. So this is the one with the completely black fur, and this is the one with the brown fur with black spots on it. The pigments involved in fur color are produced as a result of biochemical pathways that take place in cells called melanocytes. These pathways are similar to those that occur in human melanocytes. The melanocortin 1 receptor MC1R is located on the cell membrane of melanocytes and is coded by the MC1R gene. So this would be the melanocyte, and there would be a MC1R is located on the cell surface membrane of melanocytes coded for by the, so this is a protein receptor. So this would be coded by a gene for MC1R. Figure 1.3 outlines the process that occur in jaguar melanocytes. So they're giving you this. So tyrosine is converted to substance X, and then MC1R not activated, so light brown. But MC1R activated, so it will result in a black color, eumelanin and pheomelanin. So just a lot of information, new information, and this is the MC1R, that receptor, which is found in the phospholipid bilayer. So it would be something like this, and this would be the receptor found in the cell membrane. Please remember, always when we're talking of cell membrane, remember it's a phospholipid bilayer. You tend to forget that, but please remember that it's not the cell wall, which is made of cellulose. So cell surface membrane, and they've given you this. So visualization, mentally thinking, phospholipid bilayer, receptors are glycoproteins or glycolipids. Name the substance represented by X. Now, what was X here? X was tyrosine is converted to something, and then it converted to, so what is that? This is dopa, and then dopaquinone, and then melanin. So what is substance X? That was dopa. You could have said dopaquinone also, that was allowed as well. Dopa was allowed as well, dopaquinone was allowed as well, and melanin was allowed as well. So everything was allowed. Mostly, if you knew it well, you could have got away with that 1 mark. Now it says when MC1R is activated, a second messenger is produced in the cell. Give an example of a second messenger, and that's very easy. We all know that very well. Cyclic AMP. It's come in so many papers, this question. Cyclic AMP. Or you could have written it as a small c and capital A, capital M, capital P. So this got you 1 mark, this got you 1 mark. Substance X is also produced in humans, but a mutation of the TYR gene can result in substance X not being produced. So no dopa being produced, so no melanin. Now describe the phenotype of a person with this mutation. The person who doesn't have melanin looks very pale, and that phenomena is called albino or albinism. So if you said albino or albinism, well, you got a mark, but that's a very typical name. That's a biological word. So if you knew it, well, you got a mark. But if you didn't know it and you said they have a pale skin, well, fine, you would also get a mark for that. A pale skin, or if you said no pigment or no melanin in the skin, that would get you a mark as well. Then the eyes, because of the choroid, they don't have a pigment, so they have pink eyes or red eyes. And then because of this, because there's the internal refraction, choroid is the pigment which prevents internal refraction. So these people are not blind, but they'll have poor vision. So they'll have poor vision. And so this is how you write wrongly. If you say somebody is blind, what does that mean? That means he can't see. But if you say somebody has poor vision, it means he can't see very clearly. So there's a lot of difference between poor vision and blindness. And I'm sure some of you must have written blindness because it says in the mark scheme, it's a reject or ignore blindness. So red eyes or pink eyes, that got you a mark. And these three, so any 2 out of these, I'm not saying all 4 of these, any 2 points out of these would get you your 2 out of 2. And coming to the B part, the MC1R gene has 2 alleles. There are 2 alternative forms of a gene and is located on an autosome. When 2 jaguars with light brown fur mate all the offspring will have light brown fur. So light brown is what? You have to think about it. When two jaguars with black fur mate, either all of the offspring will have black fur or some will have black fur and some will have white fur. So now you know black fur is the dominant allele because they can be either this or this. But when it says light brown or all are light brown, so that means that is always the recessive trait. Because if you cross a recessive with a recessive, then all the bachas are recessive. All the offsprings are recessive. But if you cross a heterozygous dominant with another heterozygous dominant, then you can have some bachas which are recessive. So this is how you figure out the genetics questions. If it says this into this, always this, like it said in this question, light brown, always light brown. So that means light brown is changad, so changad, all changad. I'm just joking. Using symbols, construct a genetic diagram to show how two jaguars with black fur can produce some offspring with black and some with light brown fur. Now that's very easy. Now, okay, then we talked about what is an autosome. An autosome is the 22 pairs of body chromosomes. These are called autosomes. And the 23rd pair is the one which decides the gender. So if it's XX, they're equal in length, then it's XX, and that's a human being, that's a female. And if there is one in unequal size, then that's a Y chromosome. So X and Y, these are not autosomes. This is called the sex chromosome, or which decides the gender of the human being. So in this case, of course, we are talking of animals. So in animals also, some animals it is different, of course. For all, what you do is you write down the symbols. Also, capital B is for allele for black fur. This is the black fur allele, and this would be the brown fur allele. Allele for brown fur and allele for black fur. Well, you got a mark for that. This is one mark. Then the parental genotypes would be what? They would be heterozygous, big B, small b into big B, small b. Now you got that, you got one mark for that. Then you wrote this a little separately. So this is B, small b, you circled it. That's the way I like to do it. But if you didn't circle it, even then it was fine. And then you cross them. So this and this, big B, big B. Now this would be what? This would be black. Then this would be again black, but it would be heterozygous. And then again, we always write the capital first, so this would be black as well. And then of course, these would be the changars, and this would be small b, and this would be brown fur, or you can say light brown fur. So you got all your marks. You got your 4 marks. 1 mark for this symbols, 1 mark for this line, 1 mark for this line, and 1 mark for this combined line. If you did these lines, didn't matter. You did it or you didn't do it. We're not interested in the lines. We're just interested in yes, this and this. Sorry, this and this was 1 mark. Writing the gametes was 1 mark, and then this was the third mark. So 1 mark for the symbols, 1 mark for this, 1 mark for the offspring genotype, and 1 mark for the offspring phenotype. So you got your 4 marks. So I'm telling you how we mark these genetics questions. So genetics questions are those in which you can get really full marks anytime. Question number 2, photosynthesis affected by many environmental factors. Explain why light intensity can be a limiting factor in photosynthesis. 2 marks. Why do we need light? Light intensity, more light, less light. Why? Because the light energy of the photons cause the excitation of electrons for the light dependent stage. And why do we need the light dependent stage? Because it makes reduced NADP and ATP. And also light is needed to open the stomata for carbon dioxide to enter. So number 1, light energy, photons. Number 2, for the light dependent state, why do we need light energy? Why do you need sugar? Well, you want to make a cake, so you need sugar for that. Why do you want salt? You want to make an omelette, you want to add salt to it. To make NADPH and ATP and to open the stomata for the carbon dioxide to enter. You see, light is the stimulus which causes the guard cells to be activated, and the guard cells will then, of course, increase in volume and all that story, and the guard cells and the stomatal pore will open. Part 2, the concentration of carbon dioxide can also be a limiting factor. It has an effect on the Calvin cycle in the light independent stage of photosynthesis. RuBP, ribulose bisphosphate, triose phosphate, TP, and GP, glycerate 3-phosphate, are three important molecules in the Calvin cycle. Figure 2.1 shows how the concentration of GP changes when the concentration of CO2 is reduced from 0.04% to 0.008%. Reduced. Complete figure 2.1 was sketching the lines for RuBP and TP when the concentration of CO2 is reduced from 0.04 to 0.008%. So you were asked to complete this. What would happen when we reduce it from 0.004 to 0.008? What is going to happen? Okay, let's revise the Calvin cycle first. Now let's revise the Calvin cycle. In Calvin cycle, we have carbon fixation. So the RuBP combines with the carbon dioxide in the presence of the enzyme Rubisco to form a 6-carbon intermediate, which immediately breaks down. So they've not shown the 6-carbon intermediate. That breaks down to a 3-carbon, and this is called GP. Now GP is converted to TP, and it requires NADPH and ATP, right? So then the hexose sugar comes out here, and then this TP is again regenerated. So RuBP is regenerated. Now what they've said is that instead of 100 carbon dioxide, now there are only 50 carbon dioxide. So if there's only 50 carbon dioxide, that means the RuBP will now not be reacting. So there's less carbon dioxide. When there's less carbon dioxide, there'll be more RuBP because the RuBP was there previously combining 100 RuBP was combining with 100 carbon dioxide. Just giving you an example. Now they're only now 50 carbon dioxide. So 50 will combine and 50 will go free. So that is why what do? The RuBP line is going to increase. So this is what I've done. The RuBP line is going to increase and then plateau out. Well, it remain that only the 0.008% will be combining, will be combining with the RuBP to form the 6-carbon intermediate. Now, normally GP would be 100 formed if there was 100 carbon dioxide, and then there would be 100 TP. But now there will only be 50 GP and there will be 50 TP because only there is carbon dioxide is less. So the GP will be converted to, so there will be less TP now. Now let's look at the graph again. So now you can see here that the TP say was 100. Now the TP will decrease. So the TP goes down from 100, it goes down and then is less than the GP. But here also TP was less than GP. So here also the TP has to be, they've shown you GP. So it just had to be a line similar to GP because GP and TP together go together. Look at the Calvin cycle diagram. This is GP and then this is TP, which is the reduction phase in which we need the ATP and the NADPH. So just give yourself a number, say 100 were being formed when there was 0.004 carbon dioxide. Now there is 0.008 carbon dioxide, so 50 will be formed. So I'm just telling you, I'm just teaching you a way how to handle these questions. Then coming to the part 3 of the question, in very dry conditions, CO2 concentration can become the main limiting factor of photosynthesis in plants. Explain how very dry conditions cause CO2 concentration to become the main limiting factor of photosynthesis in plants. Very obvious. What is going to happen when there is going to be dry conditions? In dry conditions, there's going to be a stress response. Abscisic acid is going to be produced. Action of the calcium which acts as a second messenger, and the stomata will close. When the stomata close, then how can the carbon dioxide diffuse in? It's like you have your door closed to your room, so how can anybody enter your room? So basically what you have to go is you have to say, okay, in dry conditions, basically the stomata will close. Now this was 3 marks. Why would stomata close? Because when there is drought conditions or when there is dry conditions, a hormone called abscisic acid or a growth regulator chemical is produced, which is called abscisic acid, that causes the stomata to close. When the stomata will close, there will be no carbon dioxide diffusing into the air spaces and the palisade and reaching the chloroplast and all the stroma and everything. So you had to give me this point. This was the essential mark scheme point, no carbon dioxide diffusing, because it says very dry conditions, why it becomes the main limiting factor. So why can't carbon dioxide enter? So no carbon dioxide diffusing, but then the explanations, then you can have given me any two out of these. So you could have said stomata closed because abscisic acid is released, or because of the stress response, and it's a stress hormone produced when the plant is undergoing drought conditions. Now coming to the B part of the question, I mean, I look at all this and it gives me, I say it in Urdu, में बुखार चढ़ जाता है इतना लंबा क्वेश्चन पढ़ने से, तो ये बुखार वाला क्वेश्चन है. A factor that can limit the rate of photosynthesis is the rate of regeneration of RUBP. Pseudoheptulose 1,7-bisphosphatase, SBPase, thank you, they've given us an easy way out. SBPase is an enzyme, fine, okay, it's an enzyme in the Calvin cycle that controls the rate of regeneration of RUBP. We just did Calvin cycle. SBPase is coded for by the gene SBPase. Very nice. In an experiment, wheat plants are genetically modified to make more SBPase by introducing the SBPase gene from another grass species, Brachypodium distichyon. The resulting GM wheat plants were named SOX4. Wild type, not GM, and SOX4 plants were grown in greenhouse. So both were grown, the one which are genetically changed and the ones which were wild type, not GM, and the other one. Light intensity, CO2 concentration, and temperature were kept constant, TLC. Light intensity, temperature, and carbon dioxide concentration were kept constant. Mature plants were removed and dried to measure the biomass so that we know how much plant matter does it have. Biomass. More the biomass, more the growth. Figure 2.2 shows the mean plant biomass for the wild type plants and GM SOX4 plants. Wild type here and SOX4 here. Mean plant biomass in grams. Calculate the percentage change in mean plant biomass when these plants are grown compared to the wild type plants. Show your working. So let's look at the figures. What are the figures here? This is what? 28. And what is this? 20.5. Now, how do you calculate the percentages? Some of you chungars asked me this question so many times that it really vexes me now. 28 minus 20.5 divided by 20.5 into 100. So what will this be? So then the answer, you do the working, 7.5 over to 20.5 into 100 is equal to 36.6%. But some of you don't know the percentage change. The percentage formula is what? Final minus initial over initial into 100. Final minus initial over initial into 100. So 36.6, because it said you do it only, they didn't say anything, but you do it to one decimal place. And then coming to the part 2 of the question, it says suggest and explain why SOX4 plants have a different mean plant biomass than the wild type variety. Why? Because they have got their GM. They have an extra copy of the SBPase gene. So if they have an extra copy of the SBPase gene, that means there is more transcription of SBPase, so more SBPase will be produced. That enzyme will be more. More enzyme means more regeneration of RUBP. More regeneration of RUBP means more Calvin cycle. More Calvin cycle means more glucose being made, or more starch, or more cellulose. And also more amino acids being made, so more proteins for growth. Also there will be more cell division, more cell elongation, and more mitosis. So all that will result in, of course, more biomass. Simple. So, I mean, how do we word this? Sometimes you know it, but you don't know how to word it. So, as SOX4 have an extra copy of this SBPase gene. That was given in the question. So, there'll be more transcription of the SBPase gene. So, more regeneration of RUBP. More RUBP means more Calvin cycle. More Calvin cycle means more glucose or more starch or more cellulose. That's one mark scheme point because they're all carbohydrates. And then more amino acids for growth, and then more mitosis. More cell elongation and more mitosis. So more mitosis because the proteins and enzymes for mitosis will also be abundant. So it means that the SBPase gene was more effective. Then we come to the last part of the question. Interesting question. Some soils may be deficient in nitrates. Suggest how nitrate deficiency could limit the quantity of SVPS made by the SOX4 plants. And I couldn't think of the answer. And the answer was that, nitrogen is needed to produce nucleotides. You see, what we have to understand is that first from the DNA, which is a gene for SPPS, what has to be made? mRNA has to be made. So if you don't have the nucleotides to make this mRNA, well then how is the protein going to be made? So we needed the nitrates for the nucleotides. The nucleotides for what? For transcription. We needed the nucleotides for transcription. And what is a nucleotide? Nucleotide is RNA is a ribose sugar to which is attached a nitrogenous base. And then to this is attached a phosphate. So basically phosphate and nitrates would be needed for DNA and RNA and tRNA. Of course, I'm not saying for that, no, and also for ribosomal RNA. So, but nitrogen would be needed because they said how could the quantity of HPPS? So that means you've got to think, okay, how are proteins made? So first in protein manufacture, DNA has to be converted to mRNA. So how will the mRNA may be made if there are no nitrogenous bases? So they asked you that question. They didn't say for the cells. They just said how would it be limit the quantity? How can this enzyme? It's just like you don't. How many rotis can you make? It depends upon how much flour do you have, how much atta do you have. You only have atta for 2 rotis, so you can't make 5 rotis. So this is how this question had to be answered. Question 3 is a very direct question, and that tells you all that you must learn the cycles and all, and you must memorize them, because some direct questions is just going to ask you you're just going to check your rote learning. Aerobic respiration occurs when oxygen is available. A much greater energy yield is obtained from aerobic respiration than from respiration in anaerobic conditions. Explain why the link reaction only occurs when oxygen is available. Now, the link reaction. So, first of all, you've got to understand is that pyruvate is converted to acetyl CoA in the link reaction. And in this, NAD is needed. NAD becomes reduced, and the reduced NAD goes to ETC. And the only ETC works if oxygen is present. So what we are is why the link reaction only occurs when oxygen is available. So you had to give me the link reaction story. What happens in the link reaction? Pyruvate converted to acetyl CoA. NAD needed in the link reaction, where NAD will become reduced. NAD will become NADH. And this reduced NAD goes to ETC. But ETC does not work if oxygen is only works if oxygen is present. So ETC will only work because oxygen is the final electron acceptor. So reduced NAD is oxidized, or you can say NAD is regenerated in ETC. So how will a link reaction work if NAD is not regenerated? A little difficult this question was. So pyruvate converted to acetyl CoA. Pyruvate is converted to acetyl CoA in the link reaction. So even if you had given me the link reaction story, you'd probably have got your 2 marks at least for that. Pyruvate converted to acetyl CoA, in this NAD is needed. NAD becomes reduced. Reduced NAD goes to ETC. ETC works if oxygen is present, so NAD is regenerated. So that is why in the link reaction, number one, pyruvate does not even enter in anaerobic conditions, but it only occurs when oxygen is available. In anaerobic conditions, pyruvate will be converted to lactate or alcohol. Next, B. Outline the stage of aerobic respiration that occurs in the cytoplasm of eukaryotic cells. That's 5 marks. That's a very direct question. As I told you, you need to revise the essay questions as well. So glycolysis, phosphorylation of glucose, splitting of fructose, 1,6-bisphosphate into 2-triose phosphate, triose phosphate oxidized, net 2 ATP, and 2 reduced NAD produced. You can also write NAD, or you can also reduced NAD, and you can also write NADH. So 7 mark scheme points, and any 5 of these: glycolysis, phosphorylation of glucose, splitting of fructose 1,6-bisphosphate into 2 triose phosphates, triose phosphate dehydrogenated to pyruvate, 4 ATP produced and 2 reduced NAD, or you can say NADH produced. So reduced NAD or NADH is the same thing, you can say that. Now this was a 9-mark question, direct question. These are the questions in which you need to score, and you need to score well in these questions. So you need to really memorize all these cycles and all the basic steps in respiration and photosynthesis. Thank you. This is the video 1 completed. Question 1, 2, and 3 have been completed in this video.